Olá, pessoal. Boa tarde para vocês. É muito bom ter vocês aqui. E é com prazer que a gente vai iniciar a nossa quarta live do ciclo de palestras do PPG-RN. E para a live de hoje, a gente trouxe a convidada Guadalupe Peralta. A Guadalupe, ela é bióloga, ela é graduada pela Universidade Nacional de Córdoba, na Argentina, e é doutora em Ecologia pela University of Canterbury, na Nova Zelândia. Ela atualmente está fazendo seu terceiro pós-doutorado e em breve ela vai se ingressar como uma pesquisadora ao Conselho Nacional de Investigações Científicas e Técnicas da Argentina, que se chama de CONICET. A sua pesquisa ela foca em ecologia de comunidades e interações biológicas entre espécies. O título da apresentação dela é Species Interactions, Their Drivers and Importance. E eu, desde já, gostaria de convidar vocês a fazer perguntas ao longo de toda a apresentação, quando quer que vocês queiram, e no final a gente vai ler as perguntas e a Guadalupe vai responder. Ok, pessoal? E já peço a vocês também, para aqueles que não tiverem ainda ingressos no nosso canal do YouTube, que vocês podem fazer isso quando vocês quiserem, tá? Bom, ok, Guadalupe, creio que a gente já possa começar a nossa apresentação. E a partir de agora...
demais.
Oi?
Bom demais, eu vou compartilhar aqui.
Ok. A partir de agora é com você. Quando quer que queira, pode começar.
Aí, dá para ver a apresentação? Vocês me escutam? Estão me escutando?
Eu estou escutando sim.
Bom demais. Muito obrigada pelo convite. E vou começar com o seminário, então. So this talk is going to be, as Claudia said, about species interactions, what are the mechanisms that drive species interactions, and the importance of species interactions. So in this precise moment, I assume everyone is sitting inside in front of their computer, but if we were, if we would go outside, we would see lots of different interactions occurring among species, right? Here I just put some pictures of species interacting in different ways, right? And a lot of these interactions only last a few seconds, but they have a huge impact on our lives. So one of the most recognized examples of this are interactions between plants and pollinators. So according to the Food Agriculture Organization, approximately 75% of the world's crops that produce fruits and seeds that we consume depend, at least in part, on pollinators. So this is a huge amount of food that we rely on, and it's the type of food that we should consume more, right? Like fruits and seeds. Another example of how important or the impact that species interactions have on our lives is the example of biological control. So as you know, biological control for multiple agricultural commodities provides benefits of billions of US dollars per year, right? So this means a huge impact on our economies. And beyond these examples, I could keep going with examples, but another one that's quite interesting and intriguing to me are the interactions between plants and frugivores. So as you can see in the picture, this bird eats fruit and disperses the seeds, and the impact that these interactions have on the maintenance of our landscapes and on our forests is huge, right? Of course, these interactions, these examples I'm showing you, just two species interacting, don't occur like this in isolation, right? In communities, we have lots of different species interacting all the time in different ways. And this, the sum of all these interactions is what we call the network of interactions, right? Of the web of species interactions. And in this example of this community I'm showing you here, we have interactions occurring among plants and animals from different trophic levels. And of course, the links between the species represent their interactions. There are, in communities, there are lots of different types of interactions. We can have, for example, mutualistic interactions like plant-pollinator interactions, like the example I was showing you before as well. We could have antagonistic interactions, like herbivore-plant interactions, right? So one of the species benefits and the other one doesn't. And we could have, there are also different ways of classifying interactions. I'm not going to go into that, but there are other interactions like the frugivorous plant interactions, as I mentioned to you before. The bird eats the fruit and hopefully disperses the seeds, right? So it could be mutualistic or antagonistic if the bird doesn't disperse the seed. So the combination of interactions among a species in a community form what we call the species interactions patterns of these communities. And these interaction patterns are not only formed by the links or the occurrence of interactions, but by the frequency with which species interact. So here in this figure, the width of the link represents how frequently two species interact, right? So the wider the link between two species, the more frequently those species interact. And this is so the occurrence and the frequency of interactions is what defines species interaction patterns. And these species interaction patterns or species interactions have been shown to be affected by what we do to habitats and the landscape, right? So habitat modification. This was a seminal paper by Jason Tilianakis and collaborators that show how when we modify habitats, we are altering not only the composition of the species potentially, but also how species interact, right? And after this study, lots of different studies came comparing different types of disturbances, right? Human disturbances. So, for example, showing how deforestation affects tropical networks, and also how habitat edges, for example, also change the way in which species are interacting. How urbanization also influences species interactions, and fire events also have a huge effect on species interactions. So. What I'm going to be talking about to you today, it's basically focused on this, the species interaction patterns. So the first part of the talk is going to be about what do species interaction patterns tell us about ecosystem functions, right? So those, yes, ecosystem functions. The second part is going to be about what are the drivers or the mechanisms that allow species to interact. And the final part of the talk is going to be about what are the applied implications of an interaction network approach, of studying or using basically species interaction patterns. So let's start with the first point. What do species interaction patterns tell us about ecosystem functions? So as I was telling you before, there are lots of different interactions occurring in communities all the time. And of course, there is something like too many interactions to sample. When we go out to the field, it wouldn't be possible to sample all the interactions among all the species. It wouldn't be logistically possible, right? So that's why we tend to focus on interactions among specific groups of a species. For example, plant-pollinator interactions, right? We go and sample only the pollinators that are visiting the plant community we decide to study. Or we choose to study, for example, plant-herbivore interactions or prey interactions. In this case, we have hosts, which are caterpillars, attacked by parasitoid species, which are the predators, right? So this is a way of studying multiple species, multiple interactions among species, but at the same time being able to sample, collect data, basically, right? Oh, sorry. So it's it's been shown, or it's been suggested, that the network structure or the pattern of interactions among species in communities can be useful to understand what are the mechanisms that drive ecosystem functions. I'm going to show you an example of my PhD. So as you can see here, we have two communities, community A and community B. In both of these communities have exactly the same species composition, hosts and parasitoids, but the interaction pattern of these communities is different, as you can see. So in community A, we have a really high resource use complementarity among parasitoid species. So basically, parasitoid species feed on different hosts, and only two of them actually share one host, but the others don't. So they complement very well on how they use the resources available. On the other hand, in community B, we have that the pattern of interactions shows us that parasitoid species, all parasitoid species share at least one host with another parasitoid species, right? So how they don't complement very well among each other. Resource use complementarity in community B is very low. This this idea of how species partition resources has been suggested to be one of the mechanisms that drive the biodiversity ecosystem function relationship, right? So when we have higher biodiversity, we tend to have higher ecosystem functions. And this idea of resource use complementarity had been suggested as one of the potential mechanisms that could drive high attack rates, for example. So this is something that I decided to test during my PhD. So I collected data on hosts and caterpillars and parasitoid species in different forest types in New Zealand, native forests, plantation forests, and well, we sampled the interactions, so we could recreate the interaction pattern that occurs in the different communities we sample. And what we tested was whether resource use complementarity of parasitoid species relates to attack rates or parasitism rates of those communities. And what we found was that indeed those communities that have a higher resource use complementarity among parasitoid species also had higher parasitism rates, right? This result also later came up in different studies that use other study systems, right? So, for example, this paper came out last year from McGrath et al., and they show basically the same pattern that we found, but for interactions between plants and pollinators. So, as you can see from the figure, higher pollinator niche complementarity relates to higher seed number of seeds produced per fruit, so higher reproductive success of plants, right? And also a few years ago, Daniel Garcia's group found exactly the same pattern for plant frugivorous, plant-bird interactions, right? So they found that when birds complement in the plants they feed upon, the seed dispersal function of those communities is higher, right? So this is really interesting because it's a pattern that repeats itself on different study systems, right? And it's providing us with a mechanism, an explanation of why things change, in this case, ecosystem functions. So as a first conclusion, we could say that species interaction patterns at the community level can help us understand ecosystem functions and the changes that ecosystem functions. So, understanding how species interact, when and where species interact, it's really useful, right? It can help us, as I said before, understand changes in attack rates, in plant reproductive success, in seed dispersal. So, in order to understand, or in order to be able to answer these questions, how, when, and where species interact, we need to think about species interactions like movie scenes, right? and think, instead of focusing on the movie itself, on this movie scene we are watching, we need to go a step backwards and look at what's happening at the backstage of species interactions, right? So in this case, if we can understand why Superman is flying or how they are making him fly in the movie, we might be able to make him fly again in another movie, or at least predict when it's going to fly, right? So this brings me to the second point of the talk, which is the second part. What are the mechanisms that drive species interactions? So as you know, for an interaction to occur between two species, the co-occurrence of species is not enough. So the fact that two species are present in a certain place at a certain time doesn't necessarily mean that those species are going to interact. Species interactions also depend on lots of different factors, like the morphological traits of a species, their behavior, species physiologies, their phenologies, the abundance of species, and of course the environmental conditions in our study sites, right? When we try to predict interactions or to use these different factors to understand when an interaction is going to occur, we make usually huge assumptions that all these factors are equally important for all the interactions among all the species, right? But species have also different characteristics that could influence the importance of these different factors for the occurrence of interactions. And I'm going to show you an example of what I mean. This is a hypothetical community, so it's formed, as you can see, by plants and birds. So the birds eat the fruits of the plants. And this type of interactions, we know from previous research that for an interaction to occur between a bird and a plant, their morphology is very important. So the bill size of the bird needs to match the fruit size of the plant, and for example, the height at which the fruits are offered by the plant needs to match the height at which the bird flies, right? So we know that there needs to be trait matching between the species for an interaction to occur. However, we assume that this trait matching it's equally important for all the interactions between all birds and all plant species to occur. And this might not be the case because the species have different, as I said before, different characteristics. For example, in this community, we have exotic species and native species. And interactions among native species, or native species, for example, of birds and plants, have had a longer period of time to co-evolve. So, and we know that trait matching could be related with co-evolution. So it could be possible that interactions among native species would have a higher signal of trait matching, right, compared to interactions between natives and exotics, for example. Also, there are other characteristics from species, like their generalism degree. So, for example, in this case, the blackbird, the exotic blackbird, is a generalist, so it feeds on different plant species, compared to this other one, green. The green circle bird that is a specialist, so it only feeds on one plant species. We know as well from previous research that generalist species tend to have trait values that are more similar to the average trait values of the communities, whereas the most specialist species are those that tend to have the most extreme trait values, right? And this could influence on how important the trait matching is for the occurrence of their interactions. So we decided to test these ideas and try to answer this question: is trait matching equally important for all interactions? And in order to answer this question, we used this huge data set that George Perry, one of our collaborators, collected in New Zealand. So he basically collected, did a literature review, and collected all the information, all the interactions that occur between all the bird species and all the plant species that occur in New Zealand that have been published, right? So with this information, we could reconstruct the pattern of interactions between birds and plants in New Zealand, and we also compiled trait data of birds and plants, so that we could estimate what was the trait matching degree of the species that interact. So from this pattern of interactions, we could calculate how generalist the species are. So basically with how many partners, or how many partners they have. And so that the how many partners they have, it's on the x-axis in this figure that I'm going to show you. So in this x-axis, we have the mean generalism degree of the interacting bird and the interacting plant, right? And on the y-axis, we have how good the trait matching is between those species interacting. So what we found was that when the mean generalism degree of the species increases, so the more generalist the bird and the plant that are interacting are, the lower is the trait matching between those species, right? So this tells us that trait matching is more important for interactions that occur between specialist species, right? Another characteristic of the species that we had is whether they were native or exotic, so their biogeographic range, basically. So we tested the same idea, whether mean generalism degree and trait matching, how was this relationship, but dividing the data in the different groups of interactions depending on the biogeographic status of the interacting species. And this is what we found in all the cases. So the green line at the top represents interactions between native birds and native plants. Then the yellow one represents interactions between native plants and exotic birds, or native birds and exotic plants. And the purple one represents interactions between exotic species, exotic from New Zealand, right? It could be from anywhere in the world. And what we observed was that those interactions that occur between native species, native birds and native plants, have a higher trait matching compared to interactions that involve exotic species, right? So this happens for interactions between plants and birds, but we then started thinking, well, what would happen with in other study systems? So we decided to test the same idea with a different data set. And this one involves interactions between plants and pollinators. So in this case, this data set was collected mainly by Diego Vázquez, Nata Chakov, and Silvia Lomascolo, who you can see in the pictures, and other collaborators. So they sample plant-pollinator interactions in the Monte Desert next to the Andes in Mendoza, Argentina. And they also collected traits of the species. Of course, always when I talk about traits, are those that are involved in the interaction or in the occurrence of the interaction. For example, proboscis length of the species and corolla tube length of the flowers, right? So again, with this, with the species traits, we could estimate how was the trait matching between the species, and from the species interaction pattern at the community level, we could estimate how generalist species is, right? So with how many partners, how many flowers from different species a pollinator visits, and how many pollinators from different species a plant receives, right? And what we found was exactly the same trend as for the other data set of plant frugivorous interactions. So again, the more generalist the species that are interacting are, the lower the trait matching of those interacting species. So again, the conclusion is the same. Trait matching is more important for interactions that occur among specialist species. And Unfortunately, for this data set, we didn't have information on the biogeographic status of the species, except for, of course, Apis mellifera, the common ones. But we couldn't test the second idea with this same data set. So this is basically just a summary of what I showed you before. A specialization, as you can read in this slide, is basically the same as generalism. It's just a different name. So as you can see here, what we found was that the higher the specialization of the interacting species, the higher the trait matching, and then this influences in the occurrence of an interaction. And we decided that this was about whether an interaction occurs or not, but we decided we thought, well, what happens with the frequency of interactions, right? Because the occurrence of an interaction is one thing, and how frequently those species interact is another one. So we decided to test this idea of specialization or generalism, degree of the species, morphology, the influence of morphology on interactions, but focusing on the frequency of interactions. So in order to assess changes in the frequency of interactions, and in order to see how important morphology is for interaction frequency, we need to incorporate in our model species abundances, because the more abundant a species is, the higher the chances it's going to randomly find another species to interact. So abundance can certainly influence interaction frequency, right? So in order to be able to say how much morphology influences interaction frequency, we need to remove, in a way, the effect of abundance. So luckily for this data set of plant-pollinator interactions sample in Argentina, we had information on species abundances, so we included in this variable in our model. And species abundance can increase interaction frequency just by increasing the number of random encounters between species, but species abundance could also influence interaction frequency by increasing the phenological overlap of the interacting species. So we know also from previous research that the more abundant a species is, the longer its phenology tends to be, right? And the longer the phenology of a species, then the higher the chances that phenology is going to overlap with another species, right? So if flower, for example, stays open for a really long period of time, it has higher chances that interact with more pollinators, right? So we also had information in this data set about the phenological overlap between the different species of plants and pollinators. So we included this variable again in our model. And this is what we found. So as for interaction occurrence, we also found that the higher the specialization of a species, the higher the trait matching of the species, and the higher the interaction frequency of species. So black arrows represent here positive effects, right? But we also found that abundance has as we knew, a really strong effect on interaction frequency, but mostly driven by phenological overlap, right? So phenological overlap has a really important influence on the interaction frequency, on the frequency of interactions between plants and pollinators. And this was quite interesting. This is quite interesting because when we try to predict, for example, or assess the changes in interaction frequency, we usually think about including abundance, as I mentioned to you before. But if we include information on the phenological overlap of a species, we are incorporating the effects, most of the effects of abundance on interaction frequency, and also incorporating other characteristics of the species, right? And finally, we had some information on the pollination success of some of the plants. So we also thought, well, how would these variables influence pollination success? How would morphology or trait matching and phenology or phenological overlap influence pollination success? And what we found was that they have a positive effect, but mediated by interaction frequency. So basically, higher trait matching and higher phenological overlap generate a higher pollination success by increasing the interaction frequency of plants and pollinators. So as a second conclusion, we can say that species interaction drivers are not equally important for all interactions. And these species interaction drivers can help us understand species interaction dynamics and the changes in existing functions like pollination. And this takes me to the last part of this talk about what are the applied implications of using an interaction network approach. And to talk about this, I'm going to focus on an example of biological control and the non-target impacts of biological control. So, as you know, biological control is a tool that we use when we have, for example, a focal host that increases its abundance a lot and it's causing damage on a plant that we want to protect. So biological control is basically the introduction or the use of a biocontrol agent that we know it's going to attack our focal host and it's going to reduce its abundance, right? When we introduce a biocontrol agent, this biocontrol agent could feed on other species from the community, right? Like this other host, and could also change the abundance of those non-focal hosts, right? And this is something that we generally don't want because we want to change the community the least we need to, right? So we don't want to have this direct impact on non-target hosts. So we tend to look for the most specialist biocontrol agent to introduce, right? The one that only attacks the host that we want to control. However, even when we found the most specialized biocontrol agent, the changes in abundance of one host in the community can affect other species in the community, right? So in this case, this host, it's also attacked by two other parasitoid species, which abundances could also change. And in turn, that could cascade and translate to changes in the abundance of other species in the community, right? So these are called indirect effects via shared natural enemies. So the changes in the abundances of hosts, caterpillars that I'm showing you here. And we don't really want those to happen, right? And this doesn't only happen with biological control. It happens with any type of control or changes in the abundances that or method to control the abundance of pests, for example, that we use. We don't really want these indirect effects to happen when we use biocontrol or any measure of pest control, because these indirect effects via shared natural enemies have been shown to have a huge impact at the community level. They have been shown to drive parasitism rates at the community level, and they have been shown to determine up to 30% of the variation in herbivore abundances. So these indirect effects have a huge impact on the dynamics of the community, right? So in order to select what would be the best biocontrol agent to introduce, the one that would have the lowest direct and indirect impacts on the entire community, if we knew the pattern of interactions of the community, then we could estimate these indirect and direct effects, right? And this could be really useful to choose the best biocontrol agent. However, knowing the interactions that occur in a community, like in this example of hosts and parasitoids, it means that we need to go and sample a lot of interactions, right? And that's a huge effort, and sometimes we don't have the the money or the people to do that, right? So that's why the prediction of interactions could be really useful for assessing what would be the direct and indirect impacts of biocontrol agents. And of course, predicting interactions, it's not easy because interactions depend on all those factors that I mentioned to you before. But luckily, there are people working towards improving the prediction of interactions, and I'm going to show you an example of it. So researchers that work in models to predict interactions tend to focus on these three factors that influence the occurrence and the frequency of interactions, right? These are really, really important factors, and not saying that they are more important than behavior and physiology, but they're easier to measure than changes, for example, in behavior and physiology. That's one of the reasons why they're also more commonly used, right? So using species morphology, species abundances, and phenologies, a master student in our group, Hannah Kotula, she combined this information with machine learning approaches to try to predict what would be the direct and indirect impacts of introduced biocontrol agents. So what Hannah did was she used a different set of models. I'm going to talk about one of them only today, the random forest model. And she used data that was already available. So she used data, two data sets of host-parasitoid interactions. And these data sets contain information on species interactions, species traits, phenologies, and their abundances, right? So basically, how these machine learning approaches work is that the model learns from one of the first data set which host parasitoid trait combinations are likely to correspond to an interaction, right? So based on this, the model generates a set of rules and then applies those rules to new species, in this case from the second data set, the red one, and predict what would be the interaction probability of the new species, right, of the dataset number 2. So what Hannah thought was, well, if these interaction probabilities correspond with higher observed interaction frequencies, so if the probabilities of species interacting in the second dataset correspond to what we actually observed in the field, then we could be using these machine learning approaches to improve biocontrol risk assessment, right? And this is what she found. So in the x-axis, we have the predicted probability of 2 species interacting according to the model. And in the y-axis, we have the observed interaction frequency. So the actual frequency of interaction observed in the field in the second data set. The different colors represent different ways of training the model, or different subsets of the data set to train the model. It's not really important for the point I want to make. So, and they all have the same trend. So I'm just going to talk about them in general. So as you can see, the higher the predicted probability that the model generates corresponds with higher interaction frequencies observed in the field. So this means that this type of random forest models could be used in risk assessment to predict the attack of non-target host species and attack rates of non-target host species, right? So if we have information on which species are occurring in a community, could be information from previous studies that had been collected, and we have information on the traits, and we know the traits and some characteristics of the species we want to introduce, we can predict with a certain error, of course, but we can predict how those species are going to interact with the different species in the community we want to introduce them, So, as a final conclusion, predicting interactions can help us assess the non-target impacts of biocontrol agents, and also about other species, for example, exotic species. When exotic species is introduced, we can predict what are going to be the impacts of those exotic species in the new habitat. So, as a summary, first, species interaction patterns can provide useful information on how ecosystem functions change, and what are the mechanisms that change, that make ecosystem functions to change, right? So they're useful to understanding why things change. Second, species characteristics dictate the level of importance of the different factors that determine species interactions. So as I show you in the second section, morphological traits, for example, or trait matching is not equally important for the prediction of or the occurrence of all the interactions, but they are more important for interactions between specialist species, for example. And finally, the prediction of interactions can be used to assess non-target impacts of introduced species like biocontrol agents, exotic species, and others. Of course, these models are not perfect, and we are working towards improving them, but they are there, they are available, and people should start making use of them. And finally, thank you very much for the invitation, and thanks to all my colleagues that, of course, had a huge impact and contribution to all the work I show you. Without them, this wouldn't have been possible. And yes, I'll leave it there. Thank you.
Bom, pessoal, Guadalupe, muito obrigado pela sua palestra. Eu realmente fiquei viajando aqui nos seus conhecimentos. E a partir de agora, eu creio que a gente possa abrir para as perguntas da plateia. E com a primeira pergunta, feita pela Marina Oliveira, dentre os mecanismos que afetam as interações, a escala temporal pode ser um deles? Por exemplo, em uma determinada época, as interações podem ser mais frequentes do que em outras épocas?
Yes, absolutely. I don't know which language to answer, but I think I'll go with English. Yes, definitely. Temporal changes are very important. Those temporal changes in the interaction frequency can be driven by, for example, changes in the abundance of a species, changes in the phenology of a species, in the species availability, in the composition of a species. So yes, time it's an important it's important to consider the time step at which you are sampling, or the amount of time of data that you are compiling or combining to address your questions. So yes, time is important.
Ok, muito obrigado, Guadalupe. Então, eu acho que tem um problema com a minha conexão, mas agora voltou. Bom, a nossa próxima pergunta é do Aleph Jonathan, e ele pergunta: o fato da variável trait matching ser mais importante para espécies nativas do que exóticas tem a ver com as nativas terem evoluído juntas?
Probably, yes. That's our idea, that two species that are native have had a longer time of being together in the same place. So yes, they have probably co-evolved that degree of trait matching. So yes, it's highly possible.
Eu pensei a mesma coisa, mas fiquei em dúvida. Mas agora está respondido. Então, nossa próxima pergunta pergunta do Alexander Christianini. Ele pergunta, nice talk. Do you think interaction frequency may be used as an indirect indicator of quality of the output of the interaction for plants in other mutualistic plant-animal interactions, like frugivorous seed interactions?
It could be. It usually is, like when in a lot of the studies of plant pollinators, and I'm not quite sure about plant frugivorous, but yes, the higher the interaction frequency, the higher the functioning, or what we assume is going to be. We assume it's going to be a higher quality, as you said. So I guess there is always not like a linear relationship. There is always a point in which that quality is going to, get up to a point in which it doesn't change. Even when there are many more visits, it's going to be there's going to be a point in which the plant can't produce any more seeds, or the plant, in case of plant frugivorous birds, the plant can be dispersed to I don't know, a longer distance or stuff like that. So yes, it can be used as a quality indicator, but yes, there are certain limits, I guess, for quality.
Ok, então realmente é muito importante para a comunidade de uma forma geral. Ok, o Vitor Malin, ele pergunta como é vista a extinção local de uma espécie especialista versus uma espécie generalista em termos de perda de interações, visto que as intensidades e os tipos de interações são diferentes.
That's a very hard question to answer because it depends a lot on what your study system, I suppose. Well, if you lose a specialist species, then if there are other species also that are specialized in that species, it might have a huge effect. But so specialist species tend to have sometimes or occupy parts of the functioning niche that are more different. But at the same time, generalist species have a huge impact, right, on a lot of other species. So I guess the loss of all species, it's very it is. I mean, it's important. It's worrisome. Yes, I don't know if I'm answering your question properly, but.
Eu acho que respondeu muito bem. Bom, a próxima pergunta é do Vitor Saito, e ele fala, thank you. I recently read Peralta et al. 2020, 2020, and I was interested in how you measured neutrality with species range. Can you talk a bit about how to disentangle niche from neutrality in networks?
Yes, so for that, as I mentioned in the talk, in order to be able to say how important is morphology for the occurrence of an interaction, you need to include, or it would be ideal to include species abundances. In the case of that data set, of the one that Victor, you're mentioning, we didn't have information on the abundance of species. So we use species range as an estimate of species abundance, because the most those species that have the higher distribution range are the species that tend to be the most abundant ones, right? So we use smaller data sets to assess this idea that species range corresponds with species that are more abundant. And we found a positive relationship. So higher species range corresponds with the species that are more abundant, at least in New Zealand. So we use the species range as a substitute, as a way of abundance, right? Can you talk a bit how to disentangle niche from neutrality networks? I guess your second question, it's kind of what we try a little bit to do in that paper. So in order to assess what are the effects of the niche effects, or in our case, we use traits as a measure of niche. In order to assess how that affects species interactions and interaction networks, you need to account for neutrality, which we did with species range, right? So, yes.
Ok, muito obrigado pela resposta. Creio que tenha sido muito bem respondida. Então, a próxima pergunta é da Karine Emer. Ela fala: Hi, Lupe. Great talk. Interesting findings, especially that abundance is not that important as previously thought in determining interaction frequency. Good news because it's a difficult data to get.
Hello, Karine.
Ela pergunta a bit more on that, please.
Yes, sure. It's not that species abundance is not important, but it is important. But if we could estimate instead of abundance species phenologies or phenological overlap, then, and we include that instead of abundance, we are incorporating much of the effects abundance has on interaction frequencies. So we can substitute abundance in a way. Although I highly encourage people to sample, to collect data on species abundances independently from species interactions. But yes, that was really cool, because there are other ways in which we could estimate species phenologies and phenological overlap that might, help us when we don't have species abundance data.
Ok, muito obrigado. E a Karine tem uma outra pergunta. And also, how about interspecific variation? How strong to think is that impact on determining the generalism and specialism gradient on species interaction? Have you guys worked on that?
That's exactly what I'm about to start working on right now. Exactly that. What's the influence of intraspecific variation? The problem up to now has been that there are no or very limited data on intraspecific variation and trait variation, for example. But that question that you ask is exactly the question I'm going to be hoping to answer in the next few months or in the next year. So I'll leave the answer to that one for the next seminar.
Ok, muito obrigado, Lupe. Então, temos mais alguma pergunta para ser feita? Vou esperar um pouco pelo nosso backstage. Ok, se nós não temos, então eu vou eu mesmo fazer uma pergunta. Eu fiquei curioso para saber como é que você transforma os seus dados coletados no campo em um dado que vai responder essas perguntas a respeito das interações das espécies. É uma pergunta um pouco mais prática, da prática de campo.
When we go to the field, we sample interactions. So, for example, we observe flowers and we see which pollinator is visiting the flower, and we take notes, right? So, pollinator species X is visiting plant species J, whatever, right? So, with that data, we then create matrices. And so, basically, we have in our columns, we have for a specific site, we have the pollinator species, and the rows are plant species, and then each cell represents whether species interact or the frequency with which species interact, and that's our interaction matrix. And with that, then you can do whatever you want. There are a lot of there are a lot of metrics that have been developed, so it depends on the question you have. Well, how are you going to use the data in that in that matrix? I don't know if that's what you were asking specifically.
That's exactly. É exatamente isso que eu queria saber. Então, o Alexander, ele fez uma nova pergunta, e ele fala, I was wondering how taxonomic resolution may influence interpretation of network results. In tropical systems, the taxonomy of many groups, like insect herbivores, may be a mess.
Yes. Yeah, of course. Sorry. Parasitoid palestra or species.
Ele pergunta, how do you handle this problem in your network studies with parasitoid herbivore species? Do you believe lumping data within genera would be acceptable?
Of course, the highest or the higher the resolution you can get from your data set, the better. And for that case of parasitoid herbivore interactions, I worked a lot with taxonomists, and I think we should be encouraging more people to be taxonomists, and more funding should be driven towards keeping the taxonomy community alive. And I think it's very important to try to classify species as much as we can. Of course, there are some time limitations, and so I guess the lower the resolution, the better. But yes, if you can't identify different species from one genus, then well, that's what you can do, right? There are things that go beyond us, but so I really work a lot with taxonomists, and also I had the chance to do some DNA tests to classify species from each other, basically, or parasitoids, for example. Sometimes in some genus, the males are hard to identify, and not the females. So for some cases, we DNA barcoded males, parasitoid males, to put them together with the species they belong to, basically, and be able to use that data. So I guess those things are going to become a bit more common, like DNA barcoding and using those genetic tools. But again, I'm extremely grateful, and I admire a lot taxonomists that can actually separate things. And yes, and I think we should work more with them, and we should be encouraging more people to to take that lead.
Ok, eu acho que realmente a parte mais difícil é você ver uma abelha numa flor no meio do campo e dizer que é da espécie tal antes que ela consiga voar para longe.
Yeah, many times we actually catch the insects because it's very hard, as you said. So in those cases, we tend to collect them.
Muito obrigado. Então, eu creio que já respondemos todas as perguntas, não temos mais nenhuma, e eu gostaria de agradecer imensamente a você, Guadalupe, pela sua grande palestra, e espero também que cada um de vocês na plateia tenha gostado do que foi ensinado, do que foi compartilhado pela Guadalupe.
Well, thank you very much. Muito obrigada pelo convite.
Nós que agradecemos, Guadalupe. Bom, pessoal, só para lembrar vocês de entrar no nosso canal do YouTube e compartilhar o vídeo com as pessoas que não assistiram, não tiveram como assistir, mas que vocês gostariam de que assistissem, que vocês acharam interessante que essa pessoa veja. Então, vocês podem compartilhar com quem vocês quiserem. e também se inscrever no nosso canal. Mais uma vez, muito obrigado.